Hey there, welcome back. Get ready for molecular and cellular biology terms P through T. Stay tuned. Welcome to Science is You, a new channel dedicated to making real biotechnology research enjoyable and accessible to everyone. To support this mission, please subscribe and spread the word. Thanks for watching. P. Pseudogene. You think of the prefix pseudo like sort of. Think of the term pseudoscience. Sort of science, but not really. Similarly, a pseudogene is like a gene, but it contains a lot of mistakes and mutations, so it can't be used to make a protein anymore. Defective copies of genes. Like an instruction manual containing tons of typos and misprints, these pseudogenes were long thought of as junk DNA, but research is revealing that some of these pseudogenes might actually get copied into RNA, the intermediate step on the way to making proteins. RNA molecules themselves, though, can go on to play some roles in cell functions. Unlike pseudoscience, pseudogenes may be an important piece of the cellular puzzle. Q. Quiescence. Cells are constantly growing and dividing, following the cell cycle. But what if the cell decides to take a break? This is called quiescence. The cell retains its ability to grow and divide, but it doesn't. It just kind of hangs out. Some types of cells in the body normally reach a quiescent phase. Learning how to reactivate quiescent cells like adult stem cells to re-enter the cell cycle is an area of research. Quiescent cells can also be harmful though. For example, it may be one mechanism that allows cancer cells to escape chemotherapy treatments. Understanding cellular quiescence and its roles in the body is important for biomedical research. R. Reverse transcriptase. Proteins are like tiny complex machines made by the cell that each perform different roles. Reverse transcriptase takes molecules of RNA and copies them back into DNA. This is the reverse of what usually happens in the cell, where DNA is copied into RNA and the RNA is used to make proteins. Why would the cell want to do it backwards? This is believed to play a role in genetic diversity, allowing the cell to potentially copy and paste molecules of RNA back into DNA and integrate them in the genome. But it is also a mechanism taken advantage of by viruses, which can inject the cell with their RNA and trick them into incorporating it into the cellular genome, such as in HIV. In the lab, we can take advantage of this cellular ability by using reverse transcriptase to convert RNA that we have isolated from cells back into DNA, which is a more stable form. In this way, we can more easily study the production of different RNA molecules by cells to infer what genes are activated. S. Satellite DNA. DNA is made up of 4 molecules called bases that are used to code for the production of proteins. Satellite DNA refers to mysterious repeats in this code, where a sequence of bases appears over and over and over again in a section of the DNA called tandem repeats. These repetitive sequences don't appear to code for any proteins, but they still play important roles in DNA structure and function. T. Transgene. A transgene is a gene from one organism that has been artificially put into another organism, like a cell or mouse. This is done all the time in biotechnology research. For example, to study how the production of a certain protein inside the cell might change its behavior, we might insert an active copy of the gene into the cell. Another example is this might be done in the genetic modification of crops to help make them more resistant to disease or insects. There you have it, cellular vocabulary P through T. What do you think of these terms so far? Please share in the comments. If you enjoyed this video, please subscribe and stay tuned for the final video in this series, terms U through Z. And remember,